Hello children, welcome you all to the Form 4 biology class. Today I am going to discuss one of your second midterm content, plant nutrition, which comes under the SOW reference 2E. So during this lesson we will be discussing about the process photosynthesis, and also we are discussing about what are the factors affecting for the rate of the photosynthesis, and you will be able to understand the chemical formula for the photosynthesis and also the word equation for the photosynthesis. In addition to that, you will be able to understand that plants require mineral ions for their growth, and that magnesium ions are required for chlorophyll and nitrate ions are required for the production of amino acids. And then after we are discussing what are the deficiency symptoms which can be observed in plants. Now you can see here the word equation and the chemical formula for the photosynthesis. So basically all the green plants and green algae, they can perform this photosynthesis. Simply the photosynthesis is the process of producing complex carbohydrates utilizing carbon dioxide and water with the presence of light, and this happens within the chloroplast. Within the chloroplast, chlorophyll pigments are there. So then they are forming glucose as the major product and oxygen as the byproduct. So you should be able to remember this equation for the process photosynthesis. Actually, this is kind of like opposite of the cellular respiration. Now we will see how plants are getting the materials which are important for their growth, and how plants are getting raw materials which are required for the photosynthesis. So carbon dioxide, that is present as carbon dioxide gas in the atmosphere. So this gas diffuses into the leaf as stomata. So water is taken up by the root hairs, and then the water is transported by the xylem tissue. So the produced glucose is transported via the phloem tissue as sucrose, and the excess glucose is stored in the plant tissue as starch. This thing you have to remember. So the produced oxygen, they are diffused out of the leaves and going to the atmosphere. So basically we utilize this oxygen for our respiration. Now we will see how starch is produced and how chlorophyll is required for these photosynthesis processes. So even though plants are making this glucose via the process of photosynthesis, leaves cannot be tested for its presence, as glucose is quickly utilized. So actually this glucose is converted into starch. So in order to identify starch, we can use iodine solution. So iodine, the color is yellowish brown color. With the presence of starch, it turns into dark blue color. So this is the experiment that we can perform to identify starch which are present in the plant leaf. The first step is we are boiling this leaf in water. So why we do this? In order to break down the cell walls. Then after we are boiling this leaf in ethanol solution. So when we are boiling this leaf in ethanol, we have to use a water bath. Why? Because ethanol is extremely flammable. In order to prevent any damages and injuries, we are using a water bath here. Third step, after boiling in ethanol, we are taking off the leaf and then we are washing it using cold water. And then we have to blot it in order to remove the excess water. Then what we do is we are adding iodine solution and observing. So if the color is changing from yellowish brown to dark blue color, that means starch is present in those areas. What are the factors which are important for the photosynthesis? Light intensity, the amount of carbon dioxide, water availability, and the temperature. Right? All these factors are required for the photosynthesis. Right? So the same procedure as above can be used to investigate if light is required for the photosynthesis. So before starting this experiment, the plant required to be destarched. Destarched means removal of the starch from the plant leaves. How we are doing this? So we can perform this by placing this potted plant in a dark cupboard for about 24 hours. So this ensures that any starch already present in the leaves will be used up and it will not affect the result of the experiment. Right? So following destarching, this leaf, it can be partially covered with an aluminium foil. And then we are exposing this to sunlight. And then after we can use this leaf for the starch test. So the area of the leaf that was covered with aluminium foil, so it remains with yellowish brown color, which is the color of the iodine. So that means it did not receive any sunlight, so no starch produced in that area. But the other area which received the sunlight, so that area turned into blue-black color. So that means starch is being produced. So this proves that light is necessary for the photosynthesis. Now we will see the effect of carbon dioxide. For the photosynthesis, the same protocol can be followed. So first of all, we have to destarch the plant. Then after, we are tying a clear bag containing sodium hydroxide, or else we can use potassium hydroxide. Why we are using these chemicals? Because they are capable of absorbing carbon dioxide from the surrounding air. So the control here is a clear bag which is containing water. That is the control. For the examination, normally we can ask question, what is the control for this experiment? Right? Then you should know, okay, the clear bag which is containing water, that is the control experiment. So in which the carbon dioxide is not being absorbed. So then what we do is we are placing the potted plant in bright light for several hours. So then what we do is testing both leaves for starch. So the observations will be the bag containing sodium hydroxide or potassium hydroxide will remain orange-brown color or yellowish-brown color because it could not photosynthesize due to the absence of carbon dioxide. But in the control setup, so the color is changing from yellowish-brown to blue-black color because carbon dioxide is available, photosynthesis happen. Now we will see how to investigate the rate of photosynthesis. For this practical, we have to use an aquatic plant, especially a pondweed, right? So here what we do is, Putana, we are just observing the evolution of gas bubbles. So what is the gas produced during the photosynthesis? That is oxygen. So we can calculate the number of bubbles produced over a minute, over a unit time. So we can record these and then we can calculate the rate of the photosynthesis. So this is the experimental setup. So here you can see we are using a thermometer in order to measure the temperature. This is our pondweed. Now you can see these are the gas bubbles, oxygen gas bubbles. So we can count these using a stopwatch, right? Within a unit time, we can calculate the number of gas bubbles which have been occurred. So please check with this diagram. Investigation of the effect of changing temperature by changing the temperature of water of the beaker. So here the same protocol can be repeated by using different water baths. So we can maintain these water baths in different temperatures. So same protocol can be followed. We can count the number of gas bubbles evolved with the variation of the temperature. Alright. At the same time, we can investigate the effect of changing carbon dioxide concentration. How? By dissolving different amounts, different concentrations of sodium hydrogen carbonate or potassium hydroxide in the water. So here there's a note, special note in red color. Care must be taken when investigating a condition to keep all other variables constant in order to ensure a fair test. Only one parameter can be changed at one time. All the other parameters should be remain constant. Okay, now we will see what is a limiting factor. So most often plants do not have unlimited supplies of their raw materials, even during the photosynthesis. So temperature, light intensity, carbon dioxide concentration, and also the availability of water. These things are the main factors which can limit the rate of photosynthesis. So what is the definition for the limiting factor? So it can be defined as something present in the environment in such short supply that it restricts life processes. Now we will see the effect of temperature. Children, now check with this graph. Y-axis is the rate of photosynthesis. X-axis is the temperature. With the increment of the temperature, what happened to the rate? It has increased up to a certain point. This point, the temperature is called as the optimum temperature. Why the rate increases? Because the kinetic energy of the raw material that is increased. So there are more successful collisions that can be happen in between these reactant molecules. Then after this optimum temperature, what happens? The rate quickly drops. Why? Because enzymes are involved in these metabolic processes, so enzymes are denatured at very high temperatures. That is why this quickly drops. Now we will see light intensity. With increasing light intensity, the rate of photosynthesis increases, then it is becoming constant, kind of like leveling off, because there may be some other limiting factors. Carbon dioxide concentration, same plot puta. Now here, when we are increasing the carbon dioxide concentration, the rate of the photosynthesis also increased up to a certain point. Then after the graph is leveling off. So that means the rate becomes constant. There may be some other factors which are acting as the limiting factor because carbon dioxide is not the limiting factor. Right. Now we will see the gas exchange, which is happening in the plant body. Effect of light on net gas exchange. So normally plants are respiring all the time. So during their respiration, they are taking in oxygen and giving off carbon dioxide. At the same time, during the daytime, plants can perform photosynthesis. So what they are doing, they are just absorbing carbon dioxide and releasing oxygen to the outside environment. So there is a net intake of carbon dioxide and a net output of oxygen during the daytime. But during the night time, plants, they do not photosynthesis. Why? Because there's no light, right? But they are continuing the respiration. So that means they are taking in oxygen and giving off carbon dioxide. So always there is a net intake of oxygen and net output of carbon dioxide to the atmosphere during the night time. So we can investigate the effect of light on the net gas exchange in an aquatic plant using a pH indicator. So that is the hydrogen carbonate indicator. Now you can see these two tables, the color that the indicator turns at different levels of carbon dioxide concentration. If the concentration of carbon dioxide is highest, so what is the color change? Yellow. If it is bit high, orange. If it is in the atmospheric level, reddish color. If the carbon dioxide concentration is lower, magenta color. If it is very low, lowest, purple color, right? So now you can see here, if it is comparatively high, that means what? More respiration, less photosynthesis, lower pH. In the atmospheric level, photosynthesis equals respiration. When carbon dioxide concentration is least, so what happens? The photosynthesis is higher than the respiration, right? Okay. Come to the next table. The color that the indicator turns at different light intensities. So we can use at least 3 test tubes. So the contains are leaves, and the C test tube does not contain any leaf. What are the conditions? In test tube A, we are providing light, and even in test tube C, we provide light. But B, it's dark. So what's the color then? Indicator turns into purple color in test tube A. In test tube B, it turns into yellow color. In test tube C, it's turning into red color. So what's our conclusion? In test tube A, there is a net intake of oxygen by a leaf in light. B, there is a net intake of carbon dioxide by a leaf in the dark. C, so this is kind of like the control, right? So now we will move to the leaf structure. These are the parts of the leaf. The upper layer we call upper epidermis. Then we have palisade mesophyll, elongated cell layer. Then we have spongy mesophyll. Then after the lower epidermis. Lower epidermis contains stomata. So stomata are contained with 2 guard cells and the stomatopore. Right, children? So basically these palisade mesophyll cells, they are tightly packed. Spongy mesophyll, they are loosely packed, and that facilitates the gas exchange. And please note that upper epidermis does not contain any chlorophyll or chloroplast. But palisades and spongy mesophills, they contain chloroplast. What is the reason for this? If there are chloroplasts present in the upper epidermis, then it will be a problem for the sunlight penetration to the inner part of the leaf. That is an adaptation. That's why the upper epidermis does not contain any chloroplast. Now again, this is a cross section of the plant leaf. You can see where the stomatopores and where the intercellular spaces which are present in the spongy mesophyll, carbon dioxide can be diffused. In the same way, the produced oxygen can be diffused to the outside environment via these pores. Now we will see what are the adaptations which are present in the leaf. So leaf, it contains a waxy cuticle. So this is kind of like a protective layer which is present on top of the leaf. So this prevents the desiccation. Upper epidermis, lack of chloroplast, and it's very thin and transparent. So this is allowing the light to pass through. Palisade mesophills and spongy mesophills both are involved in the photosynthesis because they have chloroplasts. Lower epidermis, it contains guard cells and stomata. They are involved in the gas exchange. Now we will move to the guard cells. They are absorbing and losing water. This is a mechanism to open and close the stomata. Stomata is the structure which is made out of 2 guard cells and stomatal pore. Basically, this is important for the gas exchange. So during the daytime, normally the stomata are open. But during the night time, they are basically closed. Then we'll go to the vascular bundle. Vascular bundle, it contains xylems and phloem. Xylem tissue is important to transport water, while phloem tissue is important to transport food. Alright. So here, Putha, in order to maximize the rate of photosynthesis, these leaves are having a large surface area. Right? Large surface area means even if we consider a certain plant, that plant contains huge number of leaves. That is how the plant is increasing its surface area. This facilitates the light capturing, and the leaf is very thin. So this allows the carbon dioxide to diffuse to the palisades mesophyls. Presence of chlorophyll, they are important to absorb sunlight. And also network of veins. In our body we have veins, arteries and veins in order to transport substances. Likewise, within the plants they have a network of veins. Those are important to transport water and the dissolved minerals and also food particles across the plant body. Large number of stomata are present which facilitates the gas exchange. Right? Okay. Now the epidermis is thin and it's transparent. So this allows more light capturing. This thin cuticle that is actually preventing the desiccation. Not only that, it provides a protection to the plant from foreign pathogens. Okay, these palisades and spongy mesophyls, they contain chloroplast and they maximize the absorption of light. Vascular bundles, they are important for the transportation. These are the adaptations of leaf for photosynthesis. Now we will see the effects of mineral iron deficiencies. So plants obtain these elements in the form of mineral ions. So most of them are actively absorbed by the soil solution. So these plants, they have root hair cells in order to absorb these dissolved particles. Right, especially magnesium and nitrate ions. They are important for the crop. Now here you can see 3 beakers containing 3 plants. First one, what are the observations? Yellowing between veins of leaves. We call this chlorosis. And also magnesium 2 plus is required to make chlorophyll. Because of that, the plants are getting the green color. So chlorophyll needed to be absorbed light for the photosynthesis. When magnesium ions are deficient, so the leaves are getting yellow color. Second one, we can observe a healthy plant. So sufficient magnesium, sufficient nitrates. Come to the third diagram. We can observe stunted growth and yellowish leaves. So normally nitrates needed to make amino acids. Amino acids are required to produce proteins. Right? Okay. Now you studied about the functions of these magnesiums, nitrates and the deficiency symptoms. Now we will move to the questions. First question. An experiment is set up with 4 test tubes as shown in the diagram below. All 4 test tubes are left in sunlight for 6 hours. Which test tube would contain the least amount of dissolved carbon dioxide after 6 hours? Tube A contains water, tube B contains a pondweed and water with a snail. C. It contains pondweed and water. D. It contains water and snail. So which test tube would contain the least amount of dissolved carbon dioxide? Least amount of dissolved carbon dioxide. That means the carbon dioxide should be absorbed. Now in test tube A, it has just water, right? Test tube C, it has the pondweed. So when there is pondweed, it can absorb carbon dioxide. But in B and C, we can observe a snail. So during the respiration, they are producing carbon dioxide. So in B and D test tubes, there may be high amount of carbon dioxide dissolved in the water. So obviously the answer should be C. That is how normally we pick answers, right? Now I have included some more questions for you all. So you can practice these questions at home. And here, Puta, this is a link for an online test. So this test is based on MCQs. So you can just click this link and you can find the question paper. And you can try this question. And if you have any doubts, you can ask those from your teacher. That is the end of the presentation. I hope all of you enjoyed the presentation. Have a nice day.